Well, hello again. This little discussion is shorter than my previous long harangue, which will please you because it's not quite as long. But the problem I want to now address is the nature of the primordial earth. We've all learned in school that there was no oxygen in that environment. But you see, that is only an idea, because there have been no facts to show that it was correct. Well, it is correct. And now we have microbiology. We have another sulfuritum at the surface of the earth. 3.4 billion years ago. 3/4 of the age of the earth. That shows that the Earth's atmosphere was essentially devoid of oxygen. So now we know it's right, but you didn't know it was right, even though we learned that in high school. It was just an idea. It made sense. And there are lots of hypotheses that are based on it. But you see, in science, ideas are cheap. It's easy to have an idea. What costs? What takes the time? What takes the effort? What takes the creativity? Why science is so much fun is finding out whether the idea has a counterpart in reality. Is it really correct? So, this is that exercise. And I'm going to claim that the primordial earth lacked oxygen. Well, how do I know that? Well, it's based on the first discovery of what is called a microbial consortium. And you say, what is a consortium and where do they come from? Well, this particular deposit is 3.4 billion years in age. It comes from up here in northwestern Australia. And now you can see where it fits in geological time. We were talking about the modern consortium, the modern sulfuritums off of Chile, and here at 1.8 and 2.3 billion, the fossil ones, and now we're back to 3.4 billion, which is just a little bit younger than the oldest diverse assemblage of microorganisms now known in the geological record, which is about 3.5. So it's very ancient. Now we don't know much earlier than 3.5 because of what is called the geological cycle. Rocks are uplifted like in the Andes, and then they erode away and become sediments. And so those rocks no longer will exist here in another 30, 40, 50 million years. They'll be eroded away. And this goes on and goes on and goes on. So we do not have any rocks left from the origin of the Earth at 4.5 up until about 3.5, and that is when the continuous rock record begins. So this is about, at the moment, as old as we can go. Now, what is a microbial consortium? It's a fancy word. Well, it is a symbiotic, many-membered microbial community. And this is the way most microbes live today. And what does symbiotic mean? It means it's cooperative. For us humans, it's I'll scratch your back if you scratch mine. I wish that the politicians knew this so that the one side would get along with the other side so that they could compromise and help each other and thrive. But they do not. But microbes do. In this particular consortium, there are very, very different types of organisms and now live in different places. The sulfur cycling sulfuritums produce hydrogen sulfide gas, the stuff that smells like rotten eggs. And then H2S is the hydrogen source for photosynthesis in photosynthetic bacteria. Now, in cyanobacteria and in green plants, the hydrogen source is water. That's where you get the hydrogen to make the sugar in photosynthesis. But these are the first photosynthesizers. They are the simplest. And they use hydrogen sulfide. But that hydrogen sulfide has to come from someplace, and it could come from volcanoes, but they are scattered. Whereas sulfuritums at this time could live at the surface of the earth. So, I did something. Advanzo! Advanzo! Oh, makey. Do it. Okay, now we try this. Good. So, the sulfuritum, just like the ones I talked about, produces hydrogen sulfide. But if the hydrogen sulfide builds up in its immediate environment, it will kill the sulfate-reducing bacteria. It has to be used up or diffuse into the environment. And in this case, the hydrogen sulfide, which is toxic to the sulfate-reducing bacteria, is scavenged, is used by photosynthetic bacteria to make sugar. So, it is advantageous to the sulfate-reducing bacteria to get rid of the hydrogen sulfide, and it is advantageous to the photosynthetic bacteria to have a source of hydrogen for photosynthesis. So it's mutually beneficial. One is scratching the other one's back. They live together and they thrive, but they're very, very different organisms. And that is what a consortium is. Well, most organisms, most bacteria live in consortia today. And what that means is that perhaps 1%, perhaps 5% of all free-living bacteria have been cultured in the laboratory. The reason for that is that you do not know the precise growth conditions because you have just a single species. But it relies on the other species it lives with. And you don't know what they're supplying. And that's why we can't culture most living bacteria in the lab. But in nature, they live with these other things in consortia. Okay, well, this was the first consortium ever found. Why does it evidence an anoxic Earth, that is the atmosphere lacking oxygen? That's easy because the geology tells you it's living in a mud flat. It's living at the surface of the Earth, in an estuary, in a place where the water is evaporating. And in fact, you know that because it's embedded in what was a gypsum evaporite. That's where the seawater evaporates up, and when 85% of it is evaporated away, gypsum precipitates. So, the consortium was in contact with the atmosphere, and the consortium is a group that cannot stand oxygen. It's toxic. It would kill them. So that tells you there's no oxygen in the atmosphere. So how do I know that? Well, you look at the evidence. Here you see the sulfuridum swirls, these places where the sulfuridums are, and embedded with them in the same place are these laths and rosettes. And those rosettes and laths were originally gypsum. Here's one, and they were originally gypsum. They're now replaced by silica because gypsum is a salt. It dissolves away. Silica comes in and replaces it, but keeps the same crystal form. Here is another example that is 10 times younger. This is 345 million years. These are 3,400 million years, 3.5 billion years. And you can see the same morphology, the same structure, and the same things. So this is an evaporite in a mud flat. So how do you know that it's anaerobic? Well, it turns out that first place, the sulfuridums live in an anaerobic environment. But second of all, if you look at the mineralogy, you find that they are surrounded by pyrite. Pyrite is iron sulfide. Seawater sulfate gets reduced to hydrogen sulfide by the sulfuridums. There's ferrous iron, reduced iron, in that environment. It will not be present if there's oxygen. It'll be ferric iron. It'll be oxidized like rust. Okay, so it's dissolved ferrous iron. That makes pyrite, and these little pyrite balls all around the sulfuridums. So you know it's anaerobic, and you know it's living there at the surface. Oh, and some of these things, yes, they can be very nicely preserved with cells in them. And so here you can see the organic matter, the brownish stuff, that's all of these little filaments. Now, I showed you before the cobweb-like fabric of the sulfuridums that 1.8 billion, and 2.3 billion. Well, here's the same thing at 3.4 billion, where the filaments are all over the place. And they have quartz replaced gypsum within them because it's in this sort of an environment. So they clearly are sulfuridums. Now, where does the consortium business come in? Oh, what about the organisms? I showed you the organisms in the modern sulfuridum up here, the anaconda. Here's at 2.3 billion, here's at 1.8 billion, and here are the anacondas at 3.4 billion. Okay, they're not as well preserved. The organic matter has been cooked because, folks, this deposit is 1.5 billion years older than those that are 2 billion years in age. And that is very characteristic. As you go back through time, the rocks have had longer time to be cooked, to be pressure cooked by geological processes. So the organic matter is less well preserved, but it doesn't matter because you can still see the cells in them and the cell sizes and the way they're organized. Okay, well, now what about this consortium business? How do you know that? Well, it turns out that the sulfuridum bacteria, the H2S producers, are living side by side with the photosynthetic bacteria. You don't find that at the surface of the Earth today because there's oxygen in the Earth's environment, but there wasn't then. And so they cooperate. Here you can see these flat layers of the photosynthetic organisms. If they're flat like that, and they're continuous, as you can see, it means that they are in a quiet, mud-flat environment. They're not being churned up or moved. Aha! Well, now we look at the two together. The thing in the middle here, that's the photosynthetic layer. You can see the layering on it. Above it and below it, all these little balls, those are sulfuritums. So the sulfuritums and the photosynthetic bacteria are living in exactly the same environment. They're living together. That is what a consortium is. And what's going on energetically is that the sulfuridum swirls are producing the hydrogen sulfide. The hydrogen sulfide is used by the layered photosynthetic organisms to make sugar. It's the hydrogen source in photosynthesis. And that is a classic, very simple consortium. Sometimes there are 4 or 5 different organisms that contribute to various portions of the metabolic pathway. This is a simple one, but happens to be the first consortium ever discovered in the geological record because my colleagues, our paleontologists, do not know a lot about microbiology, so they don't ask the question. But then when you ask the question, the answer comes popping up. There it is. You can't miss it. Even I couldn't miss it. There it is. Aha. Okay. So we have a surface mud-flat environment, which means the organisms are in contact with the Earth's atmosphere. That environment is anoxic. The organisms are anaerobic. They do not use oxygen because there wasn't any oxygen in the atmosphere. And the whole ecosystem is anaerobic. And what that says is that there wasn't any oxygen in the Earth's atmosphere, and this is the first biological evidence of that has ever been discovered. Now others will find it because now they know that's the question to ask. But they, for some reason, didn't ask it before. So finally, I want to now look at the history of the Gallardo Espinosa sulfuridums over geological time, because we've traced them all the way back to 3.4 billion years ago. Now, to do that, what you do is you look for a modern analog, just like I did with the Black Sea and Lake Washington, to understand Metallogenium and Eoastrion, those star-shaped things that were using oxygen at the chemocline. Okay, now we look at the Persian Gulf in the United Arab Immigrants, and here is the near-shore environment. Cyanobacteria coat that environment today. So, at the present time, where we have an oxygen-rich environment, the top of the ecosystem is giving off oxygen, photosynthetic cyanobacteria, sometimes called blue-green algae. Beneath them, there are photosynthetic bacteria that are able to live because they absorb light in different parts, well, mostly in the infrared, that filters down through the cyanobacteria above. So they can still photosynthesize, but they're in an anaerobic environment. And beneath them are the sulfuridum bacteria. And beneath them, there are methane-producing bacteria and methane-consuming bacteria, methanogens and methanotrophs. So that's today in the Persian Gulf. But 3.4 billion years ago, cyanobacteria had not yet evolved. They are derived from photosynthetic bacteria, but that doesn't happen until about 3 billion years ago. So there's no oxygen being produced. So at the surface of the biota is a mixture of photosynthetic bacteria and the sulfuridum bacteria. The sulfuridum bacteria producing the hydrogen sulfide, the photosynthetic bacteria scrubbing it up, using it for photosynthesis. And then you get beneath them the sulfuridum and then the methanogens and methanotrophs, and they have gypsum laths and rosettes and pyrite, just as the modern ones do and just as the fossil ones do. Same environment, okay? But the cyanobacteria had not yet evolved. That's at 3.4 billion. At that point, the seawater sulfate is reduced to hydrogen sulfide. That's used by the photosynthetic bacteria. That's at the surface of the Earth. Now we move forward to 3 billion years. Cyanobacteria have now evolved. Now this is a real interesting thing in evolution. You see, it costs more energy. Cyanobacteria do not use hydrogen sulfide as their energy source. They use water. Now, it costs more energy to break the water molecule to get the hydrogen out costs 118 kilocalories per mole to break away that hydrogen to use it in photosynthesis. Whereas hydrogen sulfide, it's only 83 kilocalories per mole. So normally in biology, evolution goes to the more energetically efficient form. But this is the reverse. How could that be? Well, the answer is simple. The hydrogen source in water, broken off, the hydrogen reacts with carbon dioxide to make organic matter, sugar, and oxygen is given off. But oxygen is toxic to photosynthetic bacteria. It will kill them. So they have to hide underneath, get away from that oxygen. And so they do. There's this competition between cyanobacteria and the photosynthetic bacteria. The cyanobacteria win, even though it costs more energy. And what they do is it's gas warfare. They are killing off their competitors unless they can burrow underneath and hide. So cyanobacteria take over the world. And this nice consortium at 3.43 can no longer live at the surface. So what happens? Well, the sulfuridum bacteria they first get down beneath the cyanobacteria, where they sometimes live today, and then they adapt to an anaerobic environment off the shore, like they do here off the shore of Concepcion. And then over time, by 2 billion years, 2.3 and 2 billion years, they're down here into subsea muds. And they spread across the world, and so that's where they live today. And so that's the evolution of sulfuridum bacteria, back to 3.5 billion years ago. 3/4 the age of the Earth. So the take-home lesson here, folks, is that the Gallardo Espinosa Chilean sulfuridums they have spurred the discovery of the first microbial consortium. I found 3 more now, but this was the first one in 2015, I guess. And this is the first evidence of the biologic evidence, in fact, the first good evidence of the anoxic Earth that we all thought was probably true. So, folks, the take-home lesson is that sulfuridums document the history of life on this planet over billions of years. Thank you.